So my next speaker, who's right behind me, Becky Burdine, Dr. Becky Burdine, from Princeton. And she also serves on the board of directors, and she's a mom. And
That's good enough.
okay, here we go.
Sorry. Hi, everybody. Thanks for dealing with our little changes in schedule this morning. I'm going to try to catch us up a little bit. I hope you guys have enjoyed nerding out. I loved that talk because I am a science nerd. And when you hear someone give a talk about really good science, you get very excited. So I'm going to try to keep that excitement, and I'm going to tell you a little bit about ASF funding because all of you in the room are responsible for this and you need to know where your money's going. So if the talk comes up, maybe. I gave it really late, so he may be having an issue with it. No, there we go. Perfect. Okay. All right, so this is how it started for me. This is my daughter, Sophie, and she was born in 2005 and was diagnosed when she was 11 months old. And that's because she started seizing really early for a kid with Angelman syndrome. This was her in my office at Princeton because I was still working, and she would just sit there happy. I would take her to faculty meetings, and you guys are not in, those of you who aren't in academia, babies don't go to faculty meetings. So I would come in, and these really, at the time, seemed to me old men. Now they're my age, but they would like look at me and be like, "Why are you bringing the baby?" And I'm like, "What am I going to do with her?" But she was so quiet, right? And everyone would say, "What a quiet baby." And I'm like, "Yeah, I guess I'm lucky." No. Okay, so actually I am because she's absolutely fantastic. So how it started, how it's going. This is Sophie. Sophie is now 18, which blows my mind. Some of you may know me from Facebook years ago. I quit Facebook for my mental health, but she has matured into a beautiful young woman. She is delightful. She is a handful. I want to share with you one lovely thing that she has learned to do this year that we are so excited about. And that is she has learned to make a very specific noise when she needs your help. If she wants a different movie on the TV, if her toys are boring, if she is absolutely out of her mind with boredom, whatever, she makes a sound. So it's great, right? Huge increase in receptive communication and expressive communication. The problem is that sound is a pig snort, and it is so annoying. So on one hand, I'm like, yay, communication. And I'm like, please stop pig snorting. And the great thing about pig snorting is that it's funny. So if you pig snort once, laughter ensues, and then pig snorting carries on for quite a while until you fix it. Anyway, she's learned very, very smart what to do to get our attention. Okay, but this has all been a journey, right? We're all on a journey. We talk about ASF being here with you for this journey. And as you just heard, our journey started with Harry Angelman. This is him. And he reported this paper on 3 children that happened to come into his clinic that were so similar, but dissimilar to anything else he had seen. This was in Liverpool, in England. And not too many years ago, the group in Europe, they've changed their name now, but Angelman syndrome in the UK had this big gala, and it was a room like this filled with people and filled with their kids with Angelman syndrome. And they had Harry's niece come to take a prize. And she got up on the stage and she looked at all of us and she said, "I had no idea that my uncle was important to anyone." And we were all laughing, but she was like, "He was just Uncle Harry. He just was fun." She's like, "I had no idea." And it was so touching for her not to realize how important he was to so many of us. And as you just heard, we now know what causes AS. So I just want to, let me see if I can go back here. So this was in 1969 when he published this paper. And it takes a long time for things like this to sort of percolate through. But people were interested in it. More and more people were seeing kids. They were sort of putting them in the class of this Angelman syndrome that we now call it classification. And people were working on what causes AS. And this is a paper that was published by Ellen McGinnis, I think. Can't see my own slides. I don't have my glasses on. But anyway, she made the really cool finding that deletions on chromosome 15 were associated with Angelman syndrome, even though most of the time people thought they were for Prader-Willi, and it turns out it's for both. And that is a paper from Mark Lalonde's group in 1989. So we're 20 years later, 20 years down the road. And Mark's group found that if you had 2 copies of mom's chromosome 15, so uniparental disomy or UPD, for those of you who know this term, you have Prader-Willi. If you have 2 copies of dad's 15, you have this other thing, which turns out to be Angelman syndrome. So that was a huge leap forward. About 10 years later, Art Baudet, Joe Wagstaff's groups figured out that what was really important in that region of chromosome 15 was this gene we call UBE3A. And then by 10 years, actually even a year after that, once we had the gene, the first mouse models were coming out. And these were made in Art's lab by an incredibly talented geneticist named Yinghui Zhang. Ed Weber was involved in these models. Aoife Elgersma was involved in these models. All of these groups have made additional models. We got mice is covered. We've got models. This is what a mouse is. This is a knockout mouse. So we call them knockout mice because we get rid of the gene. Okay. I'm not going to belabor this because we have other sessions that are going to talk about genetics in detail, but I think this room knows that the reason you have Angelman syndrome is that you're missing a functional copy of this gene called UBE3A. And so today in Kilimanjaro 1, there's going to be Genetics 101. I'm going to give a talk on that. We'll talk about the genetics of this and what it means and what these genes are and all these names. So if you don't understand the genetics or if you're new to this journey, please show up because I love this topic. I would love to take questions and help you understand it better. At around 11:05 in the same room, there's going to be Genetics 102, and Liz Gelazzo and Katie Garabini are going to talk about how do you think about family planning and what does having that genotype mean in terms of your future. Okay, so this was sort of the roadmap to get us to where we are with the animal models. And I just want to point out that around the time we were realizing that UPD and the deletions caused Angelman syndrome, that's when the foundation was formed that Leslie talked about at the beginning. And very soon after the ASF was incorporated and formed, they started to raise money to to start to fund research. Okay, so this is the question for the audience to make sure you're awake. I have to do this to my students too, and they're in a room like this. And okay, how much research funding do you think the ASF has provided since it started funding research? So here are your choices. Is it going to be $1 million, $5 million, $15 million, or $1 billion? Yeah, I just dated myself. Okay, so you have 4 choices. So let's see some hands. How many think it's going to be $1 million? Some hands. 5 million? See some hands. Good. 15 million? Oh, wow. 1 billion? Yeah, I wish. Yeah, I wish. It's $15 million. We have funded over $15 million of research. And for a foundation that is not the National Institutes of Health, that's actually a lot of money. And what's really impressive about it is that that was raised by all of you sitting in this room, all the people that came before you. I've told plenty of researchers, like, this money is really critically important and it's precious. I mean, like, these are lemonade stands raising money so that researchers can do their research. We work really hard for this money. Our families work hard for this. We ask our friends and family. We ask them every year. We have to remind them that AS isn't over once they donate, right? We still need them to do this. It's incredibly important and we really, really appreciate it. So what do we do with your money? So this is what the philosophy has really been and still is, and what we're still working on. We do a lot of different things. So on one hand, we try to figure out what this gene UBE3A does. And you might be saying to yourself, why bother? We already have treatments. What are we doing? The reason we bother is because we could have better treatments. We don't know. And till we know what UBE3A is doing in every aspect, we can't leave that stone unturned. That is called fundamental research. There are clinical trials today, not in Angelman syndrome, but like other groups, where the whole basis of the clinical trial was because somebody thought this gene was interesting and studied it in swimming algae. If they hadn't done that, there wouldn't be clinical trials for things like ciliopathy-associated blindness. So we never know where that finding is going to come from. And that's why the fundamental research has to be the foundation. Because we are the Angelman Syndrome Foundation, and we're not just looking at fundamental research into UBE3A, we also fund things like clinical work, therapeutic endpoints, trying to find biomarkers and things so that our trials can run better. We have done a lot in clinical care and innovation. And more recently, we've been doing a lot more in this area of cutting edge therapeutics and their strategies. And this is all very important stuff. And I could spend all day. And last night I was putting slides together and I realized I was going to bore all of you to death. So what I want to do is just focus on like one or two little things just to remind you of what your work has done over the years. How do we decide what to do with that very precious money? Well, we have something called a Scientific Advisory Council, or an SAC. I'm currently the chair. It has all of these wonderful people on it. This is slide 1 and slide 2. These are all the people, and you can go to our website and look at the SAC and their little bios for everybody about what they do. But we have researchers who are currently in Angelman syndrome. We have parents who happen to have scientific backgrounds like myself and Dan Harvey and Kate Grugan. We have people from industry and finance who are really well versed in the science, who can help us make really good logical decisions. And I would name them all for you, but it'll take forever. But I'm sure you can see people up here you know. And this is the second part of that slide. So this is a large group of people. And you'll notice on here some of the clinicians that you know very well too. There was Mark Nespeka before, but Anjali is here, Ron Tebert, Win Han, and Charlie Williams. And I'm sure quite a few of you got diagnosed in this room because of Charlie. So this is a huge group. And you might be surprised to know that scientists don't get along necessarily all the time. And this is a big group of people to get to consensus, right? Because we need everybody on this panel to say, I think that's a good idea. So these meetings are incredibly fun, especially if you're nerdy. And we have to work hard at this, because if somebody says, I see a problem with that science, I don't like it, we have to discuss it and decide if we agree or disagree. Are we taking a risk? Are we not? It's a big endeavor. And so we do calls for proposals. Sometimes people come to us with stuff they think we need to do. Each proposal is assigned to 3 specific SAC members that have expertise that is relevant. Everybody reads the proposals, but those 3 people have to actually, it's like school. It's sort of bad. They all have to say, here's what I read, here's what I understood, here's what I like and don't like. And they have to say it to the rest of us, and we get to judge them. So it's really fun. All right. Then we have a meeting where we all discuss all of this. And not only do we discuss the science, because the science has to be stellar, there has to be a potential impact for our individuals with Angelman syndrome, because we can never forget that that's what we're actually doing, and that's who this is for. We score grants by this thing called the NIH system. It makes no sense because it's the government, but if it's a great grant, it gets a 1. If it's not a great grant, it gets a 9. If you're on my SAC, even though it pains me, you will put decimals into this to make it even worse. So a grant can be a 2.24. Okay, that's okay. We figure it out. And then we rank the proposals. We decide which ones we're going to recommend, and we send it to the board. And then I have to tell the board why we're recommending it. I have to defend it to the board, and the board has to agree. A lot of work goes into making sure your money is not misspent. So what have we funded? I could sit up here $15 million in 30 years and talk all day long, but I'm not going to, I promise. But let me ask you a question about this. How many of you use melatonin so that either you sleep, or you're more important, how many of you use melatonin so your individual with Angelman syndrome sleeps better? Decent number of hands in the room. Okay. Part of that reason is the very first grant we ever funded at ASF was a grant to Joe Wagstaff for him to do a small clinical trial to see if melatonin would help children with Angelman syndrome, because sleep was obviously a big issue. It turns out that it did help. And therefore, you can go to this paper, you can point to it with your physicians, and they will help you with melatonin if you need it. The first conversation I had with anybody in Angelman syndrome was Art Bodette. The second was Joe Wagstaff. And the first thing he said to me was, we just did a study. Is your child using melatonin? And I'm like, she's 11 months old. And he said, that's okay. Here's a company that makes really small doses. Give her this much. It was great, right? Clinical care changed right away. We've done other trials. You may not be aware of this because we're really hyped up now about what we're doing. But we funded two other trial approaches. One of them was a therapeutic trial for folate and betaine. Was anybody else in that study besides me? Somebody else? Dan was in it. Anybody? Somebody in the back is in it. A couple of us were in it. This was an idea that had some legs, and the idea was that if you supplemented with folate and betaine, maybe you could turn back on the paternal UBE3A copy. Yeah, spoiler, it didn't work. But we tried it. It was good science. There was a good basis for it. It was worth trying. Same thing here, levodopa, cardopa treatment for children with Angelman syndrome. There was some evidence in the community that it was working for some of the kids in the community, and so there was a trial that was done. The trial really didn't show that there was any effect for this, but that's why we do trials. Like, we do them to see if it works or if it doesn't, and sometimes they work, like the melatonin, and sometimes they don't. But that tells you what you don't have to spend your energy on. Okay. Ipa did a beautiful job talking to you about the ASO approach. I just wanted to point out that this was work that we supported from Art Bodette's group. He has been, from the get-go, from the very first conversation I had with him, he's like, we can cure this. Here's the plan. 10 years we'll be in clinical trials with something that will actually turn back on paternal UBE3A. And he was about a year off when he told me that, but he was pretty much on the money. Many of you probably know Art has a family friend. His daughter has a family friend, her closest friend, who has a child with Angelman syndrome. So Art was on this very quickly and has been committed pretty much his entire career since that point. And again, don't worry about the details here so much, but the idea is that if you use one of these things called an antisense oligo or an ASO, I'll explain it to you in Genetics 101 if you want to come, you can turn back on that copy of UBE3A that's turned off and hopefully restore UBE3A. And as you just heard, it might work really well when you're little, but it still might work pretty well when you're not. He was instrumental in getting this moving. He worked with Frank Rigo, who's on there, who's from Ionis. This approach, they've been working on this for quite a while. And we're really happy that we were there at the beginning of this. And now we have Ionis, we have Roche, we have Ultragenyx, who have all been taking this approach and doing trials. I hope you'll come to the clinical trial updates. I think is later this afternoon. They're all going to tell you about what they've done and where the programs are. I should also say thank you to everyone who has participated in these trials, because if we don't participate, we're going nowhere. I know it's hard, and I really appreciate it. You also heard about other approaches. We can put it back. We can use a virus to just put the thing back, UBE3A. These are some of the people that we have funded who've been working on that approach. And to be clear, I may be missing grants because we have a lot of them, but these are some of the more recent ones that we've done. These are all groups who are working on trying to look at viral ways of putting back UBE3A. Something we haven't talked that much about recently, but we need to start talking about again, is that maybe we can find other drugs. Betaine folate didn't work the way we wanted it to, but maybe there are some that would work. And how much better would that be than a lumbar puncture? Sorry, but like, I would love to give my child a pill. She'll swallow anything. So Dr. Philpott just published this year this paper where they have worked with a company. They've gone through another set of genes, and they're looking for small molecules or chemicals that'll turn on UBE3A. And what they're doing is what you heard about with Ipa. They're looking for neurons. So neurons are drawn like this. They look like grabby hands. And that's because neurons reach out and touch other neurons, and that's how they communicate. So in this setup, if the grabby hands are green, it's great. And if they're not green, it's not great. So if you look here, you can see that this is sort of a blankish panel. The blue shows you that they're there. There are cells there. They're just not expressing green. If you use this gene called topotecan, which is pretty harsh, it's used for cancer treatments and things like that, you can turn back on UBE3A. So very good proof of principle. This is a new chemical. And in this form of that chemical, it does as good a job, if not better, than topotecan. And so now, as of this year, we have this chemical, and this company will start to move this forward, and Ben will move this forward, and we'll see, is it dangerous? Is it toxic? How does it work? Does it turn lots of stuff on, or does it just turn on UBE3A? And it should be noted to everybody that it might be that you're going to need an ASO and you're going to need a drug to pump it up to the levels that we really need. We might need more than one therapeutic approach. So that's why you study all of them. If one works out, great. If all of them work out, even great. If we need more than one, great, we're there. All right, so I just want to give you a little bit of highlights of a meeting we just had in the last 2 days. We bring together the clinicians, the researchers, importantly, the trainees that the researchers are training up to be the next generation of researchers, because they always give the best talks. Some things in clinical work that were super exciting, and I'm sure I'm going to work with Amanda to come up with something that's a little more detailed for you so you'll have a better update of this. But we heard about evaluating infants with Angelman syndrome. So there's a group that's identifying these infants as soon as possible. So, 1-2 months. Because they sleep pretty well, they can get MRIs on sleeping infants with no anesthesia. It's incredible. They've got EEGs and they're doing sort of a time course of development. And they solved something for me that's been bothering me since the day Sophie got an MRI when she was 7 months old, because the doctor came back to me and said, Sophie has this defect in her brain. And I've always wondered, is that defect part of why Sophie is so much more affected by Angelman syndrome than most of the kids in this room? But it turns out all the infants with Angelman syndrome have that quote unquote defect. It's just a delay and it resolves. So I don't have to worry about that anymore. Guilt gone. Okay. We saw a study on how do we ask caregivers what your loved one with AS does that sort of indicates anxiety. I'm saying that in a very long-winded way instead of just saying, what does anxiety look like in Angelman syndrome? And that's because we also learned that the FDA will not accept us calling something anxiety if the person who has it can't tell you that they have it. So we have to say, yeah, this is the government. All right. So what we have to say is behaviors that look like anxiety to us in people with Angelman syndrome. Okay. But anyway, it's anxiety. They've done all these great interviews. They put together a fantastic survey and spreadsheet, and they're working out, a scale so that when we have trials, we can look and see, do these behaviors become ameliorated in the trial? Because as Aoife said, I don't need my kid to bury a marble, but it would be great if she wasn't really anxious when we went somewhere new. There was a great study from Erasmus about using this computer program to monitor how well your child receives language that only involves their eyes. So you put two things up on the screen and you say, which one is red? And the kid looks over, perfect, they scored well, right? A lot of us know our kids can do that, even if they can't do this. So it's a great measure. We also heard from various different groups working on newborn screening approaches, because we also know the sooner you get a diagnosis, the sooner you're in our family, the sooner we can get you the services that you need. Towards therapeutics, we heard an interesting talk where they are using ASO. They basically, hopefully nobody's eating. Okay, but they go into the mother mouse and they open her up. And then they go in and they inject each of the babies. She can have up 7 to 8 babies. Inject the babies while they're in utero with the ASO. Sew her back up, let her have the babies, and see like how well does it rescue Angelman syndrome. You can imagine where that's going, right? Because I've always said the most important thing for any of us is that whoever is diagnosed today walks an easier path than I did. And whoever is diagnosed 10 years from now walks an easier path than the mother today. That's really that's the goal that we should all have. And so if we could help make life with Angelman syndrome a little bit easier on you before you're even born, let's go for it. As I said, we have this new molecule that we can look at that might turn on the paternal copy. We saw all kinds of new mouse models and looking at the old mouse models to look at rescue by the re-expression and what it means. I hope that really rang for you that some of the work in the mouse models that Aoife's presented has showed that we can use these ASO approaches in people who have UPD. That's been a question for a long time, is can we do it? Can there be too much UBE3A? And the reason we ask that is we know there are conditions where if you have an additional chromosome, mother's chromosome, you have 2 mom and 1 dad, the thought is you're making too much UBE3A in addition to other genes, and that causes a completely different syndrome. So we've been worried about that. But our mouse models are telling us we don't have to worry. Everybody can be involved in these trials. And finally, the fundamental research is what I love. We've learned about how UBE3 is expressed and where it is in the cell, as you just heard. We've learned about how you regulate the expression of this gene. How much can you tolerate? We heard about mice who vocalize, and I didn't know this because we can't hear them, but mice do this ultrasonic vocalization. Like when they meet each other, they do this, beep, 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 and it makes this noise, and you can record it. And what you can see is that AS mice don't make those noises. And so they're really trying to look at what the language circuit is and why aren't they making those noises and how can we rescue it and how early do we have to do to rescue it. It's not speech, but it's a close approximation. The other one that was super cool is there is a group that is working on behaviors in Angelman syndrome, and they have this device where the mice are sitting and a food pellet comes in front of them and they have to reach out and grab the food pellet to get their bite. Very motivational. What you can see with an AS mouse, so like the normal mouse does this. He's like, okay, there's the pellet. Mice don't normally reach up, they reach down for things. So they have to learn how to do this. The AS mouse does it very slowly and then has trouble grasping the pellet. And they have been able to work out that the real problem for the motor function with this mouse is the grasping part at the end. It's that motor point at the hand. Because of that, they can go back in the brain and they can tell you exactly what region of the brain is really problematic that keeps the mouse from reaching. And the reason for this is if we can only go in and restore UBE3A to a small part of the brain, we want to pick the ones that are important. And that's one of them. That was an incredible talk. I'll try to get some movies to show you guys at a later time. All right, so incredible thanks to all of you for the funding, for the support, for sitting here and listening to me nerd out. I really appreciate it. And hopefully I've got us back on time. And if you are interested in learning more about the science and the genetics, feel free to come to my next session at 10:10 and we'll talk about genetics and why there are therapeutics on the way. Thank you so much.